Don't stop. Don't stop. Don't stop. Don't stop. Don't.
Gametogenesis, conversion of germ cells into male and female gametes. Primordial germ cells, development begins with fertilization, the process by which the male gamete, the sperm, and the female gamete, the oocyte, unite to give rise to a zygote. Gametes are derived from primordial germ cells, PGCs, that are formed in the epiblast during the second week, move through the primitive streak during gastrulation, and migrate to the wall of the yolk sac, Fig. 1. During the fourth week, these cells begin to migrate from the yolk sac toward the developing gonads, where they arrive by the end of the fifth week. Mitotic divisions increase their number during their migration and also when they arrive in the gonad. In preparation for fertilization, germ cells undergo gametogenesis, which includes meiosis, to reduce the number of chromosomes and cytodifferentiation to complete their maturation. The chromosome theory of inheritance, traits of a new individual are determined by specific genes on chromosomes inherited from the father and the mother. Humans have approximately 23,000 genes on 46 chromosomes. Genes on the same chromosome tend to be inherited together and so are known as linked genes. In somatic cells, chromosomes appear as 23 homologous pairs to form the diploid number of 46. There are 22 pairs of matching chromosomes, the autosomes, and 1 pair of sex chromosomes. If the sex pair is XX, the individual is genetically female; if the pair is XY, the individual is genetically male. 1 chromosome of each pair is derived from the maternal gamete, the oocyte, and 1 from the paternal gamete, the sperm. Thus, each gamete contains a haploid number of 23 chromosomes, and the union of the gametes at fertilization restores the diploid number of 46. Mitosis. Mitosis is the process whereby one cell divides, giving rise to 2 daughter cells that are genetically identical to the parent cell, Fig. 3. Each daughter cell receives the complete complement of 46 chromosomes. Before a cell enters mitosis, each chromosome replicates its DNA. During this replication phase, chromosomes are extremely long, they are spread diffusely through the nucleus, and they cannot be recognized with the light microscope. With the onset of mitosis, the chromosomes begin to coil, contract, and condense. These events mark the beginning of prophase. Each chromosome now consists of 2 parallel subunits, chromatids, that are joined at a narrow region common to both called the centromere. Throughout prophase, the chromosomes continue to condense, shorten, and thicken, Fig. 3a, but only at prometaphase do the chromatids become distinguishable. During metaphase, the chromosomes line up in the equatorial plane, and their doubled structure is clearly visible, Fig. 3c. Each is attached by microtubules extending from the centromere to the centriole, forming the mitotic spindle. Soon, the centromere of each chromosome divides, marking the beginning of anaphase, followed by migration of chromatids to opposite poles of the spindle. Finally, during telophase, chromosomes uncoil and lengthen, the nuclear envelope reforms, and the cytoplasm divides, Fig. 3df. Each daughter cell receives half of all doubled chromosome material and thus maintains the same number of chromosomes as the mother cell. Meiosis. Meiosis is the cell division that takes place in the germ cells to generate male and female gametes, sperm, and egg cells, respectively. Meiosis requires 2 cell divisions, meiosis I and meiosis II, to reduce the number of chromosomes to the haploid number of23, Fig. 4. As in mitosis, male and female germ cells, spermatocytes and primary oocytes, at the beginning of meiosis I replicate their DNA so that each of the 46 chromosomes is duplicated into sister chromatids. In contrast to mitosis, however, homologous chromosomes then align themselves in pairs, a process called synapsis. The pairing is exact and point for point except for the XY combination. Homologous pairs then separate into 2 daughter cells, thereby reducing the chromosome number from diploid to haploid. Shortly thereafter, meiosis, two separate sister chromatids. Each gamete then contains 23 chromosomes. Crossovers, critical events in meiosis I, are the interchange of chromatid segments between paired homologous chromosomes, Fig 4c. Segments of chromatids break and are exchanged as homologous chromosomes separate. As separation occurs, points of interchange are temporarily united and form an X-like structure, a chiasma. The approximately 30 to 40 crossovers, 1 or 2 per chromosome, with each meiotic I division are most frequent between genes that are far apart on a chromosome. As a result of meiotic divisions, genetic variability is enhanced through crossover, which redistributes genetic material. Random distribution of homologous chromosomes to the daughter cells. Each germ cell contains a haploid number of chromosomes, so that at fertilization the diploid number OF46 is restored. Polar bodies. Also during meiosis, one primary oocyte gives rise to 4 daughter cells, each with 22 plus 1 X chromosomes, Fig 5a. Only one of these develops into a mature gamete, however, the oocyte, the other 3, the polar bodies, receive little cytoplasm and degenerate during subsequent development. Similarly, one primary spermatocyte gives rise to 4 daughter cells, 2 with 22 plus 1 X chromosomes and 2 with 22 plus 1 Y chromosomes, Fig. 5b. In contrast to oocyte formation, however, all 4 develop into mature gametes. Birth defects and spontaneous abortions, chromosomal and genetic factors. Chromosomal abnormalities, which may be numerical or structural, are important causes of birth defects and spontaneous abortions. It is estimated that 50% of conceptions end in spontaneous abortion and that 50% of these abortuses have major chromosomal abnormalities. Thus, approximately 25% of concept uses have a major chromosomal defect. The most common chromosomal abnormalities in abortuses are 45X, Turner syndrome, triploidy, and trisomy 16. Chromosomal abnormalities account for 10% of major birth defects, and gene mutations account for an additional 8%. Numerical abnormalities. The normal human somatic cell contains 46 chromosomes, the normal gamete contains 23. Normal somatic cells are diploid, or 2N, normal gametes are haploid, or N-euploid refers to any exact multiple of N, example diploid or triploid. Aneuploid refers to any chromosome number that is not euploid. It is usually applied when an extra chromosome is present, trisomy, or when one is missing, monosomy. Abnormalities in chromosome number may originate during meiotic or mitotic diffulsions. In meiosis, 2 members of a pair of homologous chromosomes normally separate during the first meiotic division, so that each daughter cell receives 1 member of each pair, Fig 6a. Sometimes, however, separation does not occur, non-disjunction, and both members of a pair move into one cell, Fig 6sc. As a result of non-disjunction of the chromosomes, one cell receives 24 chromosomes, and the other receives 22 instead of the normal 23. When, at fertilization, a gamete having 23 chromosomes fuses with a gamete having 24 or 22 chromosomes, the result is an individual with either 47 chromosomes, trisomy, or 45 chromosomes, monosomy. Non-disjunction, which occurs during either the first or the second meiotic division of the germ cells, may involve the autosomes or sex chromosomes. In women, the incidence of chromosomal abnormalities, including non-disjunction, increases with age, especially at 35 years and older. Occasionally, non-disjunction occurs during mitosis. Mitotic non-disjunction, in an embryonic cell during the earliest cell divisions. Such conditions produce mosaicism, with some cells having an abnormal chromosome number and others being normal. Affected individuals may exhibit few or many of the characteristics of a particular syndrome, depending on the number of cells involved and their distribution. Sometimes chromosomes break, and pieces of one chromosome attach to another. Such translocations may be biased, in which case breakage and reunion occur between two chromosomes, but no critical genetic material is lost and individuals are normal, or they may be unbiased, in which case part of one chromosome is lost, and an altered phenotype is produced. For example, unbiased translocations between the long arms of chromosomes 14 and 21 during meiosis I or II produce gametes with an extra copy of chromosome 21, one of the causes of Down syndrome, Fig.7. Translocations are particularly common between chromosomes 13, 14.15, 21, and 22 because they cluster during meiosis. Trisomy 21 Down syndrome is caused by an extra copy of chromosome 21, trisomy 21, Fig. 8. Features of children with Down syndrome include, 1. Growth retardation. 2. Varying degrees of intellectual disability. 3. Craniofacial abnormalities, including, A. Upward slanting eyes. B. Epicondyle folds, extra skin folds at the medial corners of the eyes. C. Flat facies, and small ears. D. Cardiac defects. E. Hypotonia, Fig.9. 4. These individuals also have an increased chance of developing leukemia, infections. 5. Thyroid dysfunction. 6. And premature aging. 7. Furthermore, an increased frequency and earlier onset of Alzheimer's disease is observed among persons with Down syndrome. In 95% of cases, the syndrome is caused by trisomy 21 resulting from meiotic non-disjunction, and in 75% of these instances, non-disjunction occurs during oocyte formation. The incidence of Down syndrome is approximately 1 in 2,000 concept uses for women under age 25. This risk increases with maternal age to 1 in 300 at age 35 and 1 in 100 at age 40. In approximately 4% of cases of Down syndrome, there is an unbalanced translocation between chromosome 21 and chromosome 13, 14, 15, or 21 Fig 2.7. The final 1% is caused by mosaicism resulting from mitotic non-disjunction. These individuals have some cells with a normal chromosome number and some that are aneuploid. They may exhibit few or many of the characteristics of Down syndrome. Trisomy 18 patients with trisomy 18 show the following features: 1. Intellectual disability. 2. Congenital heart defects. 3. Low-set ears. 4. Flexion of fingers and hands, Fig 10. 6. In addition, patients frequently show micronathia. 7. Renal anomalies, syndactyly. 8. Malformations of the skeletal system. The incidence of this condition is approximately 1 in 5,000 newborns. 85% are lost between 10 weeks off gestation and term, whereas those born alive usually die by 2 months of age. Approximately 5% live beyond 1 year. Trisomy 13. The main abnormalities of trisomy 13 are, 1. Intellectual disability. 2. Holoprosencephaly. 3. Congenital heart defects. 4. Deafness. 5. Cleft lip and palate. 6. And eye defects, such as microphthalmia, anophthalmia, and coloboma, Fig 11. Asterisk. The incidence of this abnormality is approximately 1 in 20,000 live births, and more than 90% of the infants die in the first month after birth. Approximately 5% live beyond 1 year. Klinefelter syndrome. The clinical features of Klinefelter syndrome, found only in males and usually detected by 1. Amniocentesis. 2. Sterility. 3. Testicular atrophy. 4. Hyalinization of the seminiferous tubules. 5. Usually gynecomastia. The cells have 47 chromosomes with a sex chromosomal complement of the XXY type, and a sex chromatin bar body is found in 80% of cases. Bar body, formed by condensation of an inactivated X chromosome. A bar body is also present in normal females because one of the X chromosomes is normally inactivated. The incidence is approximately 1 in 500 males. Non-disjunction of the XX homologues is the most common causative event. Occasionally, patients with Klinefelter syndrome have 48 chromosomes, 44 autosomes and 4 sex chromosomes 48, XXXY. Although intellectual disability is not generally part of the syndrome, the more X chromosomes there are, the more likely there will be some degree of cognitive impairment. Turner syndrome, with a 45, X karyotype, is the only monosomy compatible with life. Even then, 98% of all fetuses with the syndrome are spontaneously aborted. The few that survive are unmistakably female in appearance, Fig 12, and are characterized by 1. The absence of ovaries, gonadal dysgenesis. 2. Short stature. 3. Other common associated abnormalities are A. Webbed neck. B. Lymphedema of the extremities. C. Skeletal deformities. D. A broad chest with widely spaced nipples. E. Approximately 55% of affected females are monosomic for the X and chromatin body negativa because of non-disjunction. In 80% of these females, non-disjunction in the male gamete is the cause. In the remainder of females, structural abnormalities of the X chromosome or mitotic non-disjunction resulting in mosaicism are the causes. Triple X syndrome, patients with triple X syndrome, 47, triple X, often go undiagnosed because of their mild physical features. However, these girls frequently have problems with speech and self-esteem. They have 2 sex chromatin bodies in their cells. Structural abnormalities, structural chromosome abnormalities, which involve 1 or more chromosomes, usually result from chromosome breakage. It has been suggested that breaks are caused by environmental factors, such as viruses, radiation. And drugs, but the evidence is inconclusive. The result of breakage depends on what happens to the broken pieces. In some cases, the broken piece of a chromosome is lost, and the infant with partial deletion of a chromosome is abnormal. A well-known syndrome, caused by partial deletion of the short arm of chromosome 5, is the Krejcik syndrome. Affected infants have 1. A cat-like cry. 2. Microcephaly, small head. 3. Intellectual disability. 4. And congenital heart disease. 5. Many other relatively rare syndromes are known to result from a partial chromosome deletion. Microdeletions, spanning only a few contiguous genes, may result in microdeletion syndrome or contiguous gene syndrome. Sites where these deletions occur, called contiguous gene complexes, are usually identified by fluorescence in situ hybridization. An example of a microdeletion occurs on the long arm of chromosome 15, 15QLL 15Q13. Note, chromosomes have a long arm, designated Q, and a short arm, designated P, based on the position of the centromere. When the microdeletion occurs on the maternal chromosome, it results in Angelman syndrome, and the children have 1. Intellectual disability. 2. Cannot speak. 3. Exhibit poor motor development. 4. And are prone to unprovoked and prolonged periods of laughter. Fig 13. If the microdeletion occurs on the paternal chromosome, Prader-Willi syndrome results. Affected individuals are characterized by 1. Hypotonia. 2. Obesity. 3. Intellectual disability. 4. Hypogonadism. 5. Undescended testes. Characteristics that are differentially expressed depending upon whether the genetic material is inherited from the mother or the father are examples of genomic imprinting. Other contiguous gene syndromes may be inherited from either parent, including Miller-Dieter syndrome. 1. Lissencephaly, 2. Developmental delay. 3. Seizures. 4. Cardiac and facial abnormalities resulting from a deletion at 17pl3, and most cases of 2ZQ11 syndrome, palatal defects. Conotruncal heart defects. Speech delay, learning disorders. And schizophrenia-like disorder. Resulting from a deletion in 22q11. Frag sites are regions of chromosomes that demonstrate a propensity to separate or break under certain cell manipulations. For example, frag sites can be revealed by culturing lymphocytes from a patient in folate-deficient medium. Although numerous frag sites have been defined and consist of CGG repeats, only those in the fMRI gene on the long arm of the X chromosome, XQ27, have been correlated with an altered phenotype that is called the Frag X syndrome. Greater than 200 repeats occur in the promoter region of the gene in affected individuals compared to 6 to 54 repeats in normal subjects. Frag X syndrome is characterized by 1. Intellectual disability, 2. Large ears, 3. Prominent jaw, 4. Large testes. The syndrome occurs in 1 per 5,000 individuals, and because it is an X-linked condition, males are affected almost exclusively, which may account for the preponderance of males among the cognitively impaired. Frag X syndrome is second only to Down syndrome as a cause of intellectual disability due to genetic abnormalities. Gene mutations. Many congenital malformations in humans are inherited, and some show a clear Mendelian pattern of inheritance. Many birth defects are directly attributable to a change in the structure or function of a single gene, hence the name single gene mutation. This type of defect is estimated to account for approximately 8% of all human malformations. With the exception of the X and Y chromosomes in the male, genes exist as pairs, or alleles, so that there are 2 doses for each genetic determinant, one from the mother and one from the father. If a mutant gene produces an abnormality in a single dose, despite the presence of a normal allele, it is a dominant mutation. If both alleles must be abnormal double dose, or if the mutation is X-linked, occurs on the X chromosome, in the male, it is a recessive mutation. Variations in the effects of mutant genes may be a result of modifying factors. The application of molecular biological techniques has increased our knowledge of genes responsible for normal development. In turn, genetic analysis of human syndromes has shown that mutations in many of these same genes are responsible for some congenital abnormalities and childhood diseases. Thus, the link between key genes in development and their role in clinical syndromes is becoming clearer. In addition to causing congenital malformations, mutations can result in inborn errors of metabolism. These diseases, among which phenylketonuria, homocystinuria, and galactosemia are the best known, may be accompanied by or cause various degrees of intellectual disability if proper diets and medical care are not instituted. Diagnostic techniques for identifying genetic abnormalities. Cytogenetic analysis is used to assess chromosome number and integrity. The technique requires dividing cells, which usually means establishing cell cultures that are arrested in metaphase by chemical treatment. Chromosomes are sustained to reveal light and dark banding patterns, G-bands, Fig 7, unique for each chromosome. Each band represents 5 to 10x10 registered base pairs of DNA, which may include a few to several hundred genes. Recently, high-resolution metaphase banding techniques have been developed that demonstrate greater numbers of bands representing even smaller pieces of DNA, thereby facilitating diagnosis of small deletions. Molecular techniques, such as fluorescent in situ hybridization, FISH, use specific DNA probes to identify ploidy for a few selected chromosomes and for detecting microdeletions. Fluorescent probes are hybridized to chromosomes or genetic loci using cells on a slide, and the results are visualized with a fluorescence microscope, Fig 15. Microarrays use spots of specific DNA sequences, probes, attached to a solid surface, usually glass or silicon. These probes may be a short sequence from a gene or other DNA element that are used to hybridize a cDNA or cRNA sample, the target sample. Hybridization of probe target sequences are detected and quantified using fluorescence or other reporter techniques. Results can detect single nucleotide polymorphisms, mutations, and changes in expression levels. Some companies now offer such techniques commercially for anyone who wants their genome tested or sequenced. Exon sequencing represents a new approach to finding mutations and polymorphisms, single nucleotide changes in a DNA sequence, responsible for birth defects and diseases. With this technique, only the coding regions, exons, in tree genome are sequenced. Together, these coding regions make up the exon and represent only 1% of the entire human genome, thereby making sequencing them more practical than trying to sequence all of the genome. Because most genetic variants lie within the coding regions for proteins, the technique is an efficient way to discover these differences. The technique is also superior to older approaches that relied on linkage studies followed by positional cloning, searching for candidate genes in specific regions of chromosomes, because these techniques required large numbers of affected individuals within a family and were not applicable to studying affected individuals from different families. In contrast, exon sequencing can find a causative mutation in a single affected individual if the exons from both parents can also be sequenced. Even sequencing affected individuals from different families regardless of kinship can be successful. It must be remembered, however, that exon sequencing can only identify variants in the coding regions of genes that alter proteins. Other genetic causes of birth defects that lie outside the coding region will have to be identified by whole genome sequencing, but for now, the expense and time required to conduct these studies is prohibitive. Morphological chances during maturation of the gametes, oogenesis. Oogenesis is the process whereby oogonia differentiate into mature oocytes. Maturation of oocytes begins before birth. Once PGCs have arrived in the gonad of a genetic female, they differentiate into oogonia, Fig 16a.b. These cells undergo a number of mitotic divisions, and by the end of the third month, they are arranged in clusters surrounded by a layer of flat epithelial cells, Figs. 17 and 18. Whereas all of the oogonia in one cluster are probably derived from a single cell, the flat epithelial cells, known as follicular cells, originate from surface epithelium covering the ovary. The majority of oogonia continue to divide by mitosis, but some of them arrest their cell division in prophase of meiosis I and form primary oocytes, Fig 16c and 17a. During the next few months, oogonia increase rapidly in number, and by the fifth month of prenatal development, the total number of germ cells in the ovary reaches its maximum, estimated at 7 million. At this time, cell death begins, and many oogonia as well as primary oocytes degenerate and become atretic. By the seventh month, the majority of oogonia have degenerated except for a few near the surface. All surviving primary oocytes have entered prophase of meiosis I, and most of them are individually surrounded by a layer of flat follicular epithelial cells, Fig 17. A primary oocyte, together with its surrounding flat epithelial cells, is known as a primordial follicle, Fig 18a. Maturation of oocytes continues at puberty near the time of birth. All primary oocytes have started prophase of meiosis I, but instead of proceeding into metaphase, they enter the diplotin stage, a resting stage during prophase that is characterized by a lacy network of chromatin, Fig 17c. Primary oocytes remain arrested in prophase and do not finish their first meiotic division before puberty is reached. This arrested state is produced by oocyte maturation inhibitor, OMI, a small peptide secreted by follicular cells. The total number of primary oocytes at birth is estimated to vary from 600,000 to 800,000. During childhood, most oocytes become atretic, only approximately 40,000 are present by the beginning of puberty, and fewer than 500 will be ovulated. Some oocytes that reach maturity late in life have been dormant in the diplotin stage of the first meiotic division for 40 years or more before ovulation. Whether the diplotin stage is the most suitable phase to protect the oocyte against environmental influences is unknown. The fact that the risk of having children with chromosomal abnormalities increases with maternal age indicates that primary oocytes are vulnerable to damage as they age. At puberty, a pool of growing follicles is established and continuously maintained from the supply of primordial follicles. Each month, 15 to 20 follicles selected from this pool begin to mature. Some of these die, whereas others begin to accumulate fluid in a space called the antennum, thereby entering the antral or vesicular stage, Fig 19a. Fluid continues to accumulate such that, immediately prior to ovulation, follicles are quite swollen and are called mature vesicular follicles or Graafian follicles, Fig 19b. The antral stage is the longest, whereas the mature vesicular stage encompasses approximately 37 hours prior to ovulation. As primordial follicles begin to grow, surrounding follicular cells change from flat to cuboidal and proliferate to produce a stratified epithelium of granulosa cells, and the unit is called a primary follicle, Fig 18 BC. Granulosa cells rest on a basement membrane separating them from surrounding ovarian connective tissue, stromal cells, that form the theca folliculi. Also, granulosa cells and the oocyte secrete a layer of glycoproteins on the surface of the oocyte, forming the zona pellucida. As follicles continue to grow, cells of the theca folliculi organize into an inner layer of secretory cells, the theca interna, and an outer fibrous capsule, the theca externa. Also, small, finger-like processes of the follicular cells extend across the zona pellucida and interdigitate with microvilli of the plasma membrane of the oocyte. These processes are important for transport of materials from follicular cells to the oocyte. As development continues, fluid-filled spaces appear between granulosa cells. Coalescence of these spaces forms the antrum, and the follicle is termed a vesicular or an antral follicle. Initially, the antrum is crescent-shaped, but with time, it enlarges, Fig 19. Granulosa cells surrounding the oocyte remain intact and form the cumulus oophorus. At maturity, the mature vesicular, graphene, follicle may be 25 mm or more in diameter. It is surrounded by the theca interna, which is composed of cells having characteristics of steroid secretion, rich in blood vessels, and the theca externa, which gradually merges with the ovarian connective tissue, Fig 19. With each ovarian cycle, a number of follicles begin to develop, but usually, only one reaches full maturity. The others degenerate and become atretic. When the secondary follicle is mature, a surge in luteinizing hormone, LH, induces the preovulatory growth phase. Meiosis I is completed, resulting in formation of two daughter cells of unequal size, each with 23 double-structured chromosomes, Fig 20 AB. One cell, the secondary oocyte, receives most of the cytoplasm, the other, the first polar body, receives practically none. The first polar body he's between the zona pellucida and the cell membrane of the secondary oocyte in the perivitelline space, Fig. 20b. The cell then enters meiosis II but arrests in metaphase approximately 3 hours before ovulation. Meiosis II is completed only if the oocyte is fertilized, otherwise, the cell degenerates approximately 24 hours after ovulation. The first polar body may undergo a second division, Fig. 20c. Spermatogenesis maturation of sperm begins at puberty. Spermatogenesis, which begins at puberty, includes all of the events by which spermatogonia are transformed into spermatozoa. At birth, germ cells in the male infant can be recognized in the sex cords of the testis as large, pale cells surrounded by supporting cells, Fig. 21a. Supporting cells, which are derived from the surface epithelium of the testis in the same manner as follicular cells, become sustentacular cells, or Sertoli cells, Fig. 2ib. Shortly before puberty, the sex cords acquire a lumen and become the seminiferous tubules. At about the same time, PGCs give rise to spermatogonial stem cells. At regular intervals, cells emerge from the stem cell population to form type A spermatogonia, and their production marks the initiation of spermatogenesis. Type A cells undergo a limited number of mitotic divisions to form clones of cells. The last cell division produces type B spermatogonia, which then divide to form primary spermatocytes, Figs. 215 and 222. Primary spermatocytes then enter a prolonged prophase, 22 days, followed by rapid completion of meiosis I and formation of secondary spermatocytes. During the second meiotic division, these cells immediately begin to form haploid spermatids. Throughout this series of events, from the time type A cells leave the stem cell population to formation of spermatids, cytokinesis is incomplete, so that successive cell generations are joined by cytoplasmic bridges. Thus, the progeny of a single type A spermatogonium form a clone of germ cells that maintain contact throughout differentiation, Fig. 22. Furthermore, spermatogonia and spermatids remain embedded in deep recesses of Sertoli cells throughout their development, Fig 2ib. In this manner, Sertoli cells support and protect the germ cells, participate in their nutrition, and assist in the release of mature spermatozoa. Spermatogenesis is regulated by LH production by the pituitary gland. LH binds to receptors on Leydig cells and stimulates testosterone production, which in turn binds to Sertoli cells to promote spermatogenesis. Follicle-stimulating hormone, FSH, is also essential because its binding to Sertoli cells stimulates testicular fluid production and synthesis of intracellular androgen receptor proteins. Spermiogenesis, the series of changes resulting in the transformation of spermatids into spermatozoa, is spermiogenesis. These changes include, 1. Formation of the acrosome, which covers half of the nuclear surface and contains enzymes to assist in penetration of the egg and its surrounding layers during fertilization, Fig 24. 2. Condensation of the nucleus. 3. Formation of neck, middle piece, and tail. 4. Shedding of most of the cytoplasm as residual bodies that are phagocytized by Sertoli cells. In humans, the time required for a spermatogonium to develop into a mature spermatozoon is approximately 74 days, and approximately 300 million sperm cells are produced daily. When fully formed, spermatozoa enter the lumen of seminiferous tubules. From there, they are pushed toward the epididymis by contractile elements in the wall of the seminiferous tubules. Although initially only SHGHTLY motile, spermatozoa obtain full motility in the epididymis. Clinical CORR elatas. Abnormal gametes in humans and in most mammals. One ovarian follicle occasionally contains 2 or 3 dearly distinguishable primary oocytes, Fig. Z5a. Although these oocytes may give rise to twins or triplets, they usually degenerate before reaching maturity. In rare cases, one primary oocyte contains 2 or even 3 nudii, Fig 25b. Such binucleated or trinucleated oocytes die before reaching maturity. In contrast to atypical oocytes, abnormal spermatozoa are seen frequently, and up to 10% of all spermatozoa have observable defects. The head or the tail may be abnormal, spermatozoa may be giants or dwarfs, and sometimes, they are joined, Fig. 25c. Sperm with morphologic abnormalities lack normal motility and probably do not fertilize oocytes. Summary: primordial germ cells, PGCs, are derived from the epiblast during gastrulation and migrate to the wall of the yolk sac in the 4th week and then to the indifferent gonad, Fig. 1, where they arrive at the end of the 5th week. In preparation for fertilization, both male and female germ cells undergo gametogenesis, which includes meiosis and cytodifferentiation. During meiosis homologous chromosomes pair and exchange genetic material. During meiosis II, cells fail to replicate DNA, and each cell is thus provided with a haploid number of chromosomes and half the amount of DNA of a normal somatic cell, Fig. 2.4. Hene, mature male and female gametes have 22 plus X or 22 plus Y chromosomes, respectively. Birth defects may arise through abnormalities in chromosome number or structure and from single gene mutations. Approximately 10% of major birth defects are a result of chromosome abnormalities, and 8% are a result of gene mutations. Trisomies, an extra chromosome, and monosomies, loss of a chromosome, arise during mitosis or meiosis. During meiosis, homologous chromosomes normally pair and then separate. If separation fails, non-disjunction, however, one cell receives too many chromosomes, and one receives too few, Fig. 6. The incidence of abnormalities of chromosome number increases with age of the mother, particularly with mothers aged 35 years and older. Structural abnormalities of chromosomes include large deletions, Cri-du-Chat syndrome, and microdeletions. Microdeletions involve contiguous genes that may result in defects such as Angelman syndrome, maternal deletion, chromosome 15QLL15QL3, or Prader-Willi syndrome, paternal deletion, 15QLL15QL3. Because these syndromes depend on whether the affected genetic material is inherited from the mother or the father, they are also an example of imprinting. Gene mutations may be dominant, only one gene of an allelic pair has to be affected to produce an alteration, or recessive, both allelic gene pairs must be mutated. Mutations responsible for many birth defects affect genes involved in normal embryological development. Diagnostic techniques for identifying genetic abnormalities include, 1. Cytogenetics to test for chromosome numbers, ploidy, 4. High-resolution metaphase banding techniques to test for small deletions. 2. Fluorescent in situ hybridization, FISH, uses fluorescent DNA probes to identify specific chromosomes or regions of chromosomes to determine deletions, translocations and ploidy. 2. Microarrays employ small sequences of DNA on chips as probes to detect mutations and changes in expression levels of specific genes. Exome sequencing sequences the protein coding region of DNA, 1% of the total DNA, the exome, to identify mutations and polymorphisms responsible for birth defects and diseases. The technique is accurate, timely, and cost-effective compared to sequencing the entire genome. In the female, maturation from primitive germ cell to mature gamete, which is called oogenesis, begins before birth. In the male, it is called spermatogenesis, and it begins at puberty. In the female, PGCs form oogonia. After repeated mitotic divisions, some of these arrest in prophase of meiosis I to form primary oocytes. By the 7th month, many oogonia have become atretic, and only primary oocytes remain surrounded by a layer of follicular cells derived from the surface epithelium of the ovary, Fig 17. Together, they form the primordial follicle. At puberty, a pool of growing follicles is recruited and maintained from the finite supply of primordial follicles.